Lecture 6 is going to be on the special senses. So this lecture is going to cover chapter 15, and it's going to go over sections 3, 5, and 6. So initially I was going to go over section 4, but I'm taking that one out. So it's only 3 sections for this chapter. So the special senses, so these are special senses that have an organ that's devoted to them. So there's vision, taste, smell, hearing, and equilibrium. Vision obviously has the eye, taste has the tongue, smell, the nose, hearing and equilibrium, the ear. All of these special senses are located on the head or in the head. Starting with the eye, so the eye has different components to it. We're not going to go over the whole anatomy of the eye because you guys should be familiar with that. But just to go over it briefly, like some of the different parts. So there's the sclera of the eye. So this is where if we follow the pathway of the light, that's where it enters first. So there's the sclera. It goes through the pupil, which is the opening. So that's right between the iris, and then it goes through the lens. Then from the lens, it's going to go to the back of the eye, which is where the retina is located. So the retina, this is where the photoreceptor cells are located. These are the cells that transduce light energy. So they get activated by the light energy. There's also neurons and glial cells located in the retina. The retina is divided into two different layers. So there's the pigmented layer and the neural layer. The pigmented layer is where you'll see the photoreceptors, and the neural layer is where you'll see the neurons, and also like the axons of the neurons. Okay, so here we can see the eye again. So here's the pigmented layer. So there's where the photoreceptors are located, and here's where they connect to those neurons. And then those neurons have axons that collect, whoops, that collect and then the form the optic nerve. So the optic nerve is the one that connects to the brain. So also in this area, so you're going to see in the area near the retina, you're going to see the optic disc. So this is the site where the optic nerve leaves the eye. And this is the area that does not have photoreceptors. Okay. So you can see photoreceptors here, photoreceptors there, but none here because of the optic nerve. So for this reason, this is called the blind spot. The retina has billions of photoreceptors, and there's two types. There's going to be rods and cones. The rods are the ones that are more light sensitive. So these are the ones that you would use at night when there's very little light. And these are mainly involved in peripheral vision. They're more numerous than the cones, but they don't provide color vision. So it's all just based on light. For the cones, these are the ones that are involved in the ability to see colors and also being able to see in high resolution. So a lot of the detail comes from the cones, and these are less numerous than the rods. They're less numerous, and these are less light sensitive. So it needs a lot more light to stimulate the cones. So in the dark, you're not using your cones, you're using the rods. In the daytime, you're using the cones and not the rods. So here you can see the rods and cones in the back. So these are located more towards the posterior side of the retina. So when the light comes in, those light rays, they hit the retina, but they go all the way to the back, and this is where they activate the photoreceptors. Once the photoreceptors are activated, they send the signal forward into the bipolar cells, and then from the bipolar cells, they send the signal towards the ganglion cells. And then here, the ganglion cells have have their axons kind of converge, and then they form the optic nerve that will exit exit the eye and go into the brain. So again, when the signal starts, I mean, when the as the light is coming in into the eye and hitting the retina, it hits the the activation starts back here when it hits the photoreceptors. So these are activated, and then they send the signal back forward to the bipolar cells, and then to the ganglion cells, and then that signal gets sent to the through the optic nerve and then to the brain. Here's another close-up of the retina. So here we look closely at the photoreceptors. So they're going to have two components. So there's the inner segment and the outer segment. The outer segment is more on the, I guess, more on the outer region of the eye. So it's closer to the outside. The inner segment would be closer to the inside. Because remember, the photoreceptors are more on the posterior or outer region of the retina, and the neurons are more towards the inside. But for the photoreceptors, there's the inner segment and the outer segment. The outer segment is where they have the photopigments, and these photopigments are embedded in discs, which we're gonna look at. If we zoom into one of these discs, we're gonna see that one of the components of these structures are little molecules, little light absorbing molecules that are called retinal, and these combine with small proteins that are called opsins. So in rods, the retinal and the opsin are going to make the the pigment rhodopsin. In cones, the retinal is going to combine with another type of protein, which you don't have to know, but they're also called the green, blue, or red proteins. And so in cones, these pigments correspond to the wavelength that stimulates them. However, some of the wavelengths do overlap. So for example, the yellow light can stimulate the red and the green cones. But if you get more red that's triggered, then you're going to see an orange color. Color blindness is when there's a lack of one or more of these cone pigments, and it's usually seen more in males. So seen in about 8 to 10% of males that have they'll have some type of color blindness, and that's because it's inherited in the X chromosome. So most of the time it's a red green color blindness, in which either the red cone or the green cones are absent. So depending on which cone is missing, uh, red can appear as green or the other way around. Phototransduction is the process by which the pigment captures a photon of light energy, and then it's converted into a graded receptor potential. So if we start, so this is very similar to the second messenger that we've already looked at. There's going to be only some slight differences, like in the enzyme that's used. But if we start here at when the light hits the pigment, okay, so here the retinal is going to absorb the light and it's going to change shape. So remember that the retinal is one of the components of the pigment. Like if it was a rod, it would be retinal and opsin. So that makes the pigment rhodopsin. But when the retinal is hit by the light, it's going to make it uh, it's going to make it change shape. And when that changes shape, it's going to change the shape of the pigment. So the light will hit the retinal, that'll change the shape of the pigment. Then the pigment is going to activate the transducin, which is the G protein here. So the G protein is then going to activate the enzyme phosphodiesterase. And that enzyme is the one that converts cyclic GMP into GMP. Cyclic GMP is cyclic guanosine monophosphate. The cyclic, or I mean, just regular GMP, that's guanosine monophosphate. So it just turns it from cyclic guanosine monophosphate to guanosine monophosphate. So when that happens, the cyclic GMP levels are going to fall, and the ion channels that are open by the attachment of cyclic GMP, those are going to start to close. And so when those channels close, sodium, in this case, sodium and calcium are not going to be able to enter the cell. So that means that the cell is not going to get those positively charged ions. So it's going to hyperpolarize. So it's going to get more negative on the inside. Photoreceptors and bipolar cells are only able to generate graded potentials. So in this case, excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Excitatory postsynaptic potentials, those are the ones that cause depolarization. Inhibitory postsynaptic potentials, they cause hyper uh, polarization. Okay, but they do not generate a action potential. But when light hyperpolarizes the photoreceptor cells, they're going to stop releasing the inhibitory neurotransmitter. So in this case, it's glutamate to the bipolar cells. And then the bipolar cells are no longer going to be inhibited. So they're going to depolarize. And then they're going to release the neurotransmitter into the ganglion cells, and the ganglion cells are going to generate the action potential to the brain, or that's going to lead to the brain. So if we look at what happens in the dark, you have the cyclic GMP gated channels that are open, and that's allowing a cation influx. So positive charge ions are able to go in. So that means that the photoreceptor depolarizes. Then the voltage-gated calcium channels are going to open in the synaptic terminals. The neurotransmitter is going to be released. That neurotransmitter is going to cause the inhibitory postsynaptic potential in the bipolar cell, so here, and that's going to cause hyperpolarization. Hyperpolarization is going to cause the voltage-gated calcium channels to inhibit the neurotransmitter. So that means that no excitatory postsynaptic potential is going to occur in the ganglion. And then since there's no excitatory postsynaptic potential, there's going to be no action potentials that occur in the optic nerve. In in light, in the presence of light, the opposite happens. So here we have the cyclic GMP channels that are going to be closed. So there's going to be um, no influx of positive charged ions. Okay, so that means that the photoreceptor is going to hyperpolarize because there's no positive ions going in. The voltage-gated calcium channels are going to be closed in the synaptic terminals. So no neurotransmitter is going to be released. And because there's no neurotransmitter, there's going to be a lack of inhibitory postsynaptic potentials in the bipolar cells. So that's going to cause depolarization. And then depolarization is going to cause the voltage-gated calcium channels to open, and then the neurotransmitter is going to be released. When those neurotransmitters are released, the excitatory postsynaptic potentials occur in the ganglion. And then in that case, the action potentials are generated and they are propagated through the optic nerve. So for light and dark adaptations, light adaptations are when you move from a darkness into bright light. You're going to see a glare because the rods and cones are going to be strongly stimulated. And so this is going to cause your pupils to contract. And so they're going to get narrower to limit or to reduce the amount of light that goes in through your pupils. So it usually improves in about 5 to 10 minutes as the rod system is going to turn off and the retinal sensitivity is also going to decrease. For dark adaptation, that's when you move from bright light into darkness. So here the cones are the ones that stop functioning. at low light, because remember these are less sensitive to light. The cones are less sensitive to light, so they need a lot of light to be activated. So at low light, they'll stop functioning. So in this case, the pupils will dilate to so that the eyes will be able to take in more light or any available light. And here the rhodopsin, which is the pigment, is going to accumulate in the dark, and the retinal sensitivity is going to start to increase. Remember, retinal was the molecule. And then the transducin is going to return to the outer segments. And then sensitivity is going to increase within about 20 to 30 minutes. Okay, so I'm splitting this video into two parts. So I'll put up the smell and taste video next.